Thank you for that introduction, Kevin. So the title of my dissertation is New Perspectives on Primary Production in the Pacific Sector of the Southern Ocean. To introduce my dissertation work, I'm going to first give you some background on the topics that I'll touch on throughout this talk. So the view on the map that I'm showing you here on the right is as if you picked up the globe and looked at the bottom with Antarctica in the center and the southern tip of South America, Africa, and Australia visible around the margins. When we talk about the Southern Ocean, we're talking about the part of the world's oceans that I'm showing you here, which is the ocean south of 40 degrees south. The Southern Ocean is an incredibly dynamic system with huge fluctuations in sunlight, temperature, and sea ice over the course of a year. To illustrate that point, I'm showing you the maximum winter sea ice extent in 2020 on the left and the minimum summer extent a little over 4 months later in February of 2021 on the right. The difference between the maximum and minimum sea ice extent in the Southern Ocean is over 6 million square miles. Essentially, the Southern Ocean gains and loses the entire Arctic wintertime sea ice area every year. By contrast, the difference between wintertime and summertime sea ice extent in the Arctic is only about 3.5 million square miles. My work focuses on net primary production, or NPP, in the Southern Ocean. Net primary production is the net uptake of carbon by photosynthetic organisms. We talk about net primary production because photosynthetic organisms use some of the carbon that they take up in photosynthesis for cellular processes, and we're interested in how much carbon they retain. On land, primary producers are grasses and trees and plants. But in the Southern Ocean, primary producers are single-celled photosynthetic organisms called phytoplankton. An example species is shown in the background of this slide. Because they are single-celled, the movement of these organisms in the ocean is dictated by the movement of the water that they are in, which is why we call them plankton. This is in contrast to organisms like fish or whales, which move themselves through the water. So when we talk about net primary production in the Southern Ocean, we're talking about the net uptake of carbon by single-celled photosynthetic plankton. Now, I'm again showing you that bottom of the globe view, but this time the data on the map, aside from the land masses, are showing primary production. Areas that are red or orange show high production, and areas that are blue or purple are low production, and green is kind of in the middle. Now, all of this production takes place during a pretty short time period in the austral summer, which is from around November to March, as the sun rises and the sea ice rapidly retreats. What I want you to take away from this image is that primary production in the Southern Ocean does not occur uniformly throughout the region. And my focus is on what controls this non-uniformity and determines the patterns and productions that we see in the Southern Ocean. We care about what phytoplankton are doing, where they're growing, how much carbon they're taking in for two major reasons. The first reason is that phytoplankton play a major role in the global carbon cycle. As we dump billions of metric tons of carbon into the atmosphere each year, it has become a pressing issue to understand the details of how the global carbon cycle functions so that we can predict the impacts of the inevitable global warming that results from these carbon emissions. Phytoplankton are an important part of that cycle, and the Southern Ocean, through the action of phytoplankton, is responsible for 30% of global carbon export in the world's oceans, despite the Southern Ocean comprising only about 6% of global ocean surface area. Can you guys hear my dog whining outside the door? Sorry, it's distracting me. Just want to make sure no one else can hear it.
Nope, can't hear it.
Okay. Okay. So this carbon export process begins with carbon dioxide dissolved in the ocean at equilibrium with atmospheric carbon dioxide concentrations. Phytoplankton take up this dissolved carbon dioxide and turn it into biomass using sunlight as a source of energy. That phytoplankton biomass then has the potential to sink, taking that carbon with it to deep water masses that won't interact with the atmosphere again for hundreds of years. Or even to sediments where that carbon may not make it back to the atmosphere for millions of years. This phytoplankton-mediated transport of carbon dioxide from the surface ocean to carbon stores in the deep ocean is known as the biological pump. Without the action of the biological pump in the oceans, atmospheric carbon dioxide concentrations would be almost double their present-day values. The second reason we care about primary production is that phytoplankton form the base of the Southern Ocean food web, with fish, seals, penguins, and whales all relying on phytoplankton production either as their food source or their food source's food source. While this may look complicated, compared to many other ecosystems, the Southern Ocean is a relatively simple food web with few trophic links. If we look only at the food chain of baleen whales, for example, it looks more like this. Baleen whales feed on krill, which feed on phytoplankton. This is important because it means that changes in primary production directly impact upper trophic level organisms like baleen whales, seabirds, seals, and penguins. For my dissertation work, I've focused on the Pacific sector of the Southern Ocean. I've outlined my study region in red here, and I'm going to talk about why I focused on this region specifically next. So now I'm showing you a slightly different view of the world, with the Pacific Ocean more or less in the middle, North America on the top, Asia on the top right, and Asia on the top left, and my study region outlined in blue. The data on this map shows the trend in air temperature between present-day air temperature and average air temperature between 1951 and 1980. The darker the red, the more warming a region has experienced. Generally speaking, this map is just alarmingly red. However, my study region in the blue box shows almost no increase in air temperature, despite warming trends immediately to the east and west. The Pacific sector of the Southern Ocean is one of the few places on the planet that is showing little to no warming in air temperature. When we look at the ocean, we can see that this region is actually cooling. Now I'm showing you a map of, again, that bottom of the globe view of the Southern Ocean. This map shows the trend in sea surface temperature since 1979, with red indicating warmer temperatures and blue indicating colder temperatures. My study region is outlined in magenta. What I want you to take away from this figure is that despite a broad warming trend across much of the Southern Ocean, we are seeing a significant cooling trend in sea surface temperature in the Pacific sector of the Southern Ocean and in my study region. We also see this trend in sea ice. This map is now showing the trend in sea ice in the Southern Ocean, with blue indicating an increase in sea ice and red indicating a decrease in sea ice, with my study region again outlined in magenta. In addition to decreasing sea surface temperatures, we are also seeing a significant increase in the persistence of sea ice in my study region. Since 1979, the length of the sea ice season, which is the number of days a region is under the ice, has increased by almost 2 months. The overarching question behind my dissertation work is what drives the patterns of net primary production that we observe in the Pacific sector of the Southern Ocean. My dissertation is divided into 3 chapters, each of which looks at a different aspect of this question. In my first chapter, I examine the impact of the changes in sea ice and sea surface temperature that I've just described on net primary production, and this chapter was published in the Journal of Geophysical Research Oceans in 2016. My second chapter focuses on an area of unexpectedly high production from my first chapter and was published in Nature Communications this past February. My final chapter looks at the interannual variability in the high productivity region from chapter 2 and is in preparation for submission to the Journal of Geophysical Research Oceans. I will only be presenting results from my first 2 chapters today. So, chapter 1. Excuse me. As I've just mentioned, my first chapter examines the impact that the changes in sea ice and sea surface temperature observed over the last few decades in the Pacific sector of the Southern Ocean have had on net primary production. To do this, I conducted a satellite-based study of net primary production, sea ice, and sea surface temperature. Net primary production is calculated as a function of phytoplankton abundance, sea surface temperature, and sea ice cover, and light. Let's not forget light, all of which we measure using satellites. The most fundamental data for this calculation is the estimate of phytoplankton abundance in the water. And this measurement is done by estimating the amount of chlorophyll A in the water, which is the primary photosynthetic pigment used by both phytoplankton and land plants. It is essentially what makes plants green, and that is more or less how we measure it. So this is a true color image from the MODIS Aqua satellite, which is the satellite we use to get our chlorophyll data. This image is from the Ross Sea. Most of the white stuff you see here is ice or snow on land. Some of it's sea ice, the wispier looking stuff is clouds, but I want to draw your attention to the green stuff. in the water. This is a phytoplankton bloom. At a basic level, this is how our satellite chlorophyll algorithms determine the amount of chlorophyll in the water by looking at the different wavelengths of light measured by the satellite and estimating how green the water is and using that to calculate chlorophyll concentration. So using satellite estimates of chlorophyll as opposed to going out there and measuring it ourselves gives us amazing spatial and temporal data coverage. What I'm showing you here are global satellite estimates of ocean chlorophyll. And unlike the previous image, these are colored to show chlorophyll concentration, with red and yellow indicating high chlorophyll and blue and purple low chlorophyll. So every single day, we get an image like the ones on the left. And it's kind of hard to make out clear details in these images. But every 8 days, we can put the data from the daily images together to get an image like the one on the right. In this image, you can clearly see the shape of continents. Essentially, except for regions of persistent cloud cover, we get a global estimate of chlorophyll A every 8 days. And satellite data is also amazing as a long-term record. So when I developed my research question, I immediately had access to more than 15 years of satellite data so that I could really look at long-term trends. So first, I want to reorient you to my study region. The larger figure shows a detailed map of the study region, and the inset map in the upper right shows the position of this region relative to Antarctica. I want you to note that I've rotated Antarctica by 180 degrees from the views that I have shown previously. And New Zealand is pretty much straight north now. This is so that when we look at the detailed map, north is up and south is down. Looking at the detailed map on the left, I would like to draw your attention to a few features. First, the red line, which shows the shelf break, which is the 1,000 meter depth line. And this line divides the shallow continental shelf to the south, called the Ross Sea, with depths shallower than 1,000 meters, from the deep off-shelf region to the north of the line, which has depths closer to 4,000 meters. I also want to point out the dotted purple line towards the top of the figure, which shows the average position of the winter sea ice edge. So next, I'm going to walk you through some of the more descriptive results from this study to give you a better sense of the region. So this figure shows the average number of open water days per year from 1992 to 2013 across the study region. And we see the highest number of open water days in red, close to the edge of the winter sea ice extent, indicated by the dotted line at the top of the figure. The number of open water days increases as we move south, first changing to yellow, then green, with some areas of persistent sea ice shown in blue and purple. When we move onto the shelf, we actually start to see an increase in the number of open water days. And this is because strong winds push the sea ice away from the Antarctic coast, creating areas of open water very early in the year for such a southern latitude. So this figure shows average annual sea surface temperature for 1992 to 2013. And this shows a similar pattern to what we just observed in open water days, with the highest sea surface temperatures in red to the north and a gradual decrease in sea surface temperature, first to yellow, then to green, as we move south. We see the lowest sea surface temperatures, the purples and dark blues, in the areas of persistent sea ice cover. And we also see higher sea surface temperatures on the shelf corresponding with the area where we saw a high number of open water days. This figure shows average annual net primary production from 1997 to 2013 in grams of carbon per meter squared per year. Net primary production is much higher on the shelf than in the off-shelf region. Net primary production on the shelf ranges from around 60 to 125 grams of carbon per meter squared per year, while the off-shelf region is mostly under 25 grams of carbon per meter squared per year. But because the shelf is small compared to the off-shelf region, when you integrate spatially, which is to say you take production rate per square meter and multiply it by the number of square meters, the balance shifts and the off-shelf region really dwarfs production on the shelf. This distinction is important because areas of high productivity like the Ross Sea shelf are important concentrations of resources for upper trophic level organisms such as seals, whales, and penguins. However, even small changes in net primary production in the off-shelf region can have a huge impact on carbon cycling simply because it's such a large region. So here I'm showing the change in mean annual sea surface temperature with time. The colors indicate the slope of the relationship between sea surface temperature and time. Blue is a negative relationship, so colder temperatures with time, and red is a positive relationship, warmer temperatures with time. And areas of statistical significance are outlined in black. Now I've added a figure showing the relationship between open water days and time. Red is an increase in the number of open water days with time, and blue is a decrease. Looking at the change in sea surface temperature and open water days with time, we see a large region of cooling sea surface temperatures and a decrease in the number of open water days across the majority of the off-shelf portion of the study area, and a region of warming sea surface temperatures and an increase in the number of open water days along the eastern margin of the study region. Here. This figure shows the change in net primary production with time. Again, blue is a decrease with time and red is an increase with time. What I'd like you to see is that the pattern and the trend in net primary production shown here is very similar to what I just showed you for sea surface temperature and open water days, with a large area of decrease across the off-shelf region and a small area of increase along the eastern margin. Now I'm showing you the relationship between net primary production and sea surface temperature on the left, and the relationship between net primary production and open water days on the right. Red indicates a positive relationship and blue a negative relationship. And areas of significance are again outlined in black. As you can see, net primary production shows a significant positive relationship with both sea surface temperature and open water days across the majority of the southern portion of the study region. This relationship of net primary production with sea surface temperature and open water days is likely driving the changes in net primary production with time that I've just shown you. Now I'd like to talk about interannual variability in sea surface temperature, open water days, and net primary production, and how this interannual variability is impacted by variations in atmospheric circulation. I'm interested specifically in how the southern annular mode, or SAM, impacts interannual variability in these variables. The southern annular mode is a climate index. The climate index you're probably best acquainted with is the El Niño Southern Oscillation Index, which describes the patterns in wind over the tropical Eastern Pacific Ocean. The SAM index is similar to that, but for the Southern Hemisphere. So next, I'm going to give you some background on the southern annular mode and then show you some more results. The southern annular mode describes the pressure gradient from high pressures in the mid-latitudes to low pressures in the high latitudes. During a negative phase of the SAM, shown on the left, this pressure gradient is not as strong, and during a positive phase of the SAM, this pressure gradient is stronger. This pressure gradient drives the westerly winds that circle Antarctica. The stronger pressure gradient during a positive SAM relative to a negative SAM results in an increase in westerly wind intensity and a poleward contraction in the position of the westerly winds. The Ross Sea responds very strongly to the southern annular mode because of the effect the southern annular mode has on the semi-permanent low pressure system adjacent to the Ross Sea, shown here in orange, called the Amundsen Sea Low. This low pressure system drives meridional, which is north-south winds in the Pacific sector, resulting in cold southerly winds in the Ross Sea coming from the Antarctic continent. During a positive SAM, this low pressure system deepens and the southerly winds blowing into the Ross Sea intensify. This figure shows the state of the southern annular mode since the mid-1950s, with time on the x-axis and the value of the southern annular mode on the y-axis. The state of the SAM is highly variable, ranging from about -4 to +4, with the majority of values falling between -2 and +2. I also want you to note that the southern annular mode shows a positive trend over time. The figure on the left is showing sea surface temperature versus the southern annular mode, and the figure on the right is showing open water days versus the southern annular mode. And red indicates a positive relationship, and blue is a negative relationship. And what you can see is that the shelf and off-shelf portions of the Ross Sea behave completely opposite in response to the state of the southern annular mode, both in the response of sea surface temperature and the response of open water days to the state of the SAM. In response to a positive SAM, we see an increase in sea surface temperature and an increase in open water days on the shelf, and a decrease in sea surface temperature and a decrease in open water days off the shelf. The changes in open water days in response to the SAM are very large. In response to a 1 unit increase in the SAM, we will see a 15-day increase in open water days across a large part of the shelf. The SAM tends to fluctuate between -2 and +2, which yields a 60-day swing in open water days on the shelf across the range of the SAM. And this figure shows the relationship between net primary production and the southern annular mode. You can see that the pattern in the response of net primary production to the southern annular mode is similar to that of sea surface temperature and open water days, with a decrease in net primary production across a large portion of the off-shelf area and an increase in net primary production on the shelf in response to a positive southern annular mode. And this makes sense considering the results that I showed you earlier of the significant positive relationship between net primary production and sea surface temperature and net primary production in open water days. The increase in net primary production on the shelf in response to a 1 unit increase in the SAM ranges from about 15 to 30 grams of carbon per meter squared per year. The average annual rate of production on the shelf ranges from around 60 to 125 grams of carbon per meter squared per year. What this means is that a 1 unit increase in the state of the SAM can yield a 25% increase in production on the shelf. So the major points that I want you to take away from this work are that we see decreased sea surface temperatures and open water days over time in the study region. And this has resulted in a decrease in net primary production across a large portion of the study region. I also want to emphasize the opposite response of the shelf and off-shelf portions of the study region to the southern annular mode in terms of sea surface temperature, open water days, and production. These findings have implications for the carbon cycle in the region. We have seen a significant decrease in net primary production in the off-shelf portion of the study region since 1997, meaning that phytoplankton are taking up less carbon. However, with the positive trend in the southern annular mode and the influence that the state of the SAM has on this region, it is possible that increases in production on the shelf may eventually balance the decreases observed off the shelf. These findings also have implications for resource availability for upper trophic level organisms such as penguins. To illustrate this point, I've chosen an example from the literature that looks at the overwintering positions of 2 colonies of Adelie penguins that both breed on the same island in the Ross Sea. This figure is from a study by Ballard et al. In 2010 and shows the overwintering positions of Cape Crozier penguins in orange and Cape Royds penguins in blue. In the summer, both of these penguin colonies breed on Ross Island, indicated by the orange and blue dots at the bottom of the figure. And then they migrate to their respective overwintering areas. The figure I've just added on the right shows net primary production versus the southern annular mode. The blue and orange dots indicate the summertime positions of both penguin colonies. The blue box indicates the position of the Cape Royds penguins during the winter, and the orange box, the position of the Cape Crozier penguins during the winter. What I'd like you to see here is that while these penguins will experience similar conditions in the summer during their breeding season, due to the proximity of their breeding grounds, during a strong positive southern annular mode, they could experience very different conditions in their overwintering ranges. During a strong positive SAM, the Cape Royds penguins could encounter waters that had been more productive during the previous summer than the Cape Crozier penguins to the east. And this could actually result in the 2 colonies having a different population response to the southern annular mode, which means that the patchy response of net primary production in this region to the southern annular mode is an important context in which to view resource availability for the upper trophic level organisms that feed throughout this region. Okay, chapter 2. So the title of chapter 2 is a bit of a spoiler, so I'm not going to remind you what it was. Instead, I'm going to frame this section in the way that it developed. And it started with this very basic question. What is going on with this area of high production in the middle of what is supposed to be a low productivity region? You may remember from the results that I've just shown you that the shelf is a high productivity area and the off-shelf region is low productivity. So So what's going on here? Second. So now I'm showing you a different map of mean annual net primary production. We've zoomed out a bit, but we're still in the same region. The inset map in the top right corner shows the position of the large figure relative to the Antarctic continent. Just to get you reoriented, the Ross Shelf bloom, which we discussed in chapter 1, is indicated by the orange arrow. And the area of elevated productivity that I'm interested in is indicated by the pink arrow. This area of elevated net primary production falls within the boundaries of the Antarctic circumpolar current, shown by the dashed white lines. The Antarctic circumpolar current flows from west to east and is driven by the westerly winds. It is the dominant circulation feature in the Southern Ocean and the largest current in the world in terms of the amount of water that it moves. It's also generally an unproductive region in the Southern Ocean. However, the phytoplankton bloom indicated by the pink arrow is persistent in the same general location year after year, enough so that it appears on an image showing a 22-year average of annual net primary production. Now here's a look at that same bloom from a few different years. Please note that I've zoomed in on the bloom region from the previous view that I was showing you, and I again look to the inset map for orientation. So each of these panels shows annual net primary production during a different year. The bloom in 2009-2010 on the top right was very large and highly productive, while the bloom in other years, for example, 2018-2019 on the bottom right, was smaller and less productive. Across all of the years shown here, you can get a sense for the fact that the bloom returns to the same spot every year, though some years are stronger than others. And this interannual variability in productivity between years is actually the focus of my third chapter. Sorry, I keep ducking out of view. My nose decided to start running. So that's why I keep disappearing. In January of 2014, we were fortunate to sample this area of elevated production during a research cruise to the region. So now I'm going to show you some of those results. The results we're going to look at next are zoomed in on this pink box, and the inset map in the top right will help keep you oriented. This figure shows a composite satellite chlorophyll image from November of 2014 through November of 2013 through February of 2014, spanning the time that we were there collecting samples in January of 2014. The chlorophyll image is overlaid with our cruise track. It's a bit hard visually to get a sense of the size of this bloom, but in 2013-2014, the bloom was about 266,000 square kilometers, which is about half the size of the state of California. It's big. So next, I'm going to show you data from samples that we collected in the water column along a transect indicated by the pink line. The transect will go from west on the left to east on the right, same as the line. Now that figure that I just showed you is in the top right-hand corner of the slide. The two sections shown below are heat maps based on the concentrations of chlorophyll on the left and nitrate on the right, measured in the water column. Our sampling locations and depths are indicated by the dots. Both sections are down to a depth of 100 meters. As a reminder, chlorophyll is an indicator of phytoplankton abundance, and nitrate is an essential nutrient needed for phytoplankton growth. It's the same stuff that you put on your garden to help your plants grow. Chlorophyll concentrations within the bloom were much higher than outside the bloom. 6 milligrams of chlorophyll per meter cubed in the bloom versus 0.6 outside of the bloom. And the growth of the phytoplankton indicated by the elevated chlorophyll concentrations resulted in nitrate concentrations being drawn down to 15 micromolar in the bloom, as opposed to 30 micromolar outside of the bloom. I want to draw your attention to the fact that while nitrate concentrations were drawn down in the bloom by the growth of phytoplankton, they had not been drawn down to 0. And the Southern Ocean is known for this phenomenon of high concentrations of nitrate remaining in surface waters. So here I'm showing you a map of surface nitrate concentrations in the world's oceans. And while the majority of the ocean shows concentrations of nitrate close to 0 in surface waters, the Southern Ocean has concentrations in excess of 20 micromolar. And in case you're having difficulty finding the Southern Ocean on this map, it's the giant orange blob at the bottom of the figure. This is because the growth of phytoplankton in the Southern Ocean is limited by the availability of iron rather than nitrate, which is the nutrient that limits phytoplankton growth for most of the rest of the world's oceans. In the majority of the ocean, iron is abundant enough to not limit phytoplankton growth. And this is because the largest source of iron input to the oceans is from dust swept off of continents by the wind and deposited in the ocean. This satellite picture shows an example of dust being swept off the coast of the African continent and into the Atlantic Ocean. But because Antarctica is almost entirely covered by ice, there is very little dust to be picked up by the wind, and therefore very little dust is deposited in the Southern Ocean. So on the left, I am now showing a section of chlorophyll down to a depth of 400 meters, and on the right is dissolved iron concentration to the same depth. Iron was depleted in surface waters by phytoplankton growth, both inside and outside the bloom. But as you can see, we found elevated dissolved iron concentrations under the bloom. Iron concentrations below 100 meters under the bloom were around 0.33 nanomolar, while iron concentrations below 100 meters outside the bloom were around 0.15 nanomolar. Given that phytoplankton growth is limited by iron in the Southern Ocean, the presence of iron under the bloom is most likely the mechanism behind the presence of the bloom. So now on the left, I'm showing you the same section of dissolved iron concentrations that was on the previous slide, as well as dissolved iron concentration profiles down to 2000 meters for the stations indicated by the inverted triangles above the section. Profiles from bloom stations are shown in green, and profiles from non-bloom stations are in gray. What you can see is that the dissolved iron concentrations at bloom stations remained elevated relative to the deep profile at our non-bloom station down to almost 2000 meters. Again, I believe the bloom is present because of the elevated iron concentrations. This figure shows vertical iron delivery on the x-axis and phytoplankton abundance on the y-axis at each station, and this is calculated for each station that we sampled. So we calculated the delivery of iron from 100 meters to surface waters and found a significant positive relationship between the delivery of iron to surface waters and the abundance of phytoplankton. And this is to say, again, that the iron under the bloom is the likely cause of the bloom. So then we're left with the question, where did all this iron come from? The bloom returns to the same spot every year. The bloom is possible due to the presence of elevated iron. So the iron must also return to the same spot every year. So this figure is looking at the part of the Southern Ocean south of the Indian Ocean, with the bottom of Australia visible in the top right of the map. The data show the average chlorophyll concentration from 1998 to 2007. Because sediments are a major source of iron in the Southern Ocean, recurring blooms like the one I've been describing within the boundaries of the Antarctic circumpolar current are associated with shallow features such as islands and shallow plateaus, where iron-rich sediments can be easily swept up into surface waters. An excellent example of this is the bloom that happens downstream of the Kerguelen Islands each year, shown above, where you can clearly see the elevated chlorophyll plume downstream of the islands. However, there is no island or plateau in the region of the bloom that I'm studying. So again, where is the iron coming from? Now, this figure shows bottom depth in the Pacific sector of the Southern Ocean. The Antarctic continent is visible along the bottom of the figure, and the continental shelf of New Zealand is visible at the top in the center. Now I've added the chlorophyll image of the bloom from 2013-2014, the year that we were there sampling. The position of the bloom image relative to the larger bottom depth image is shown by the yellow box. While the bloom that we've been talking about is not near an island or a plateau, it does fall above a mid-ocean ridge, which is a seafloor mountain system formed at the boundary between tectonic plates where seafloor spreading is taking place. And this brings an intriguing possibility for the source of our mystery iron into play. Mid-ocean ridges, indicated by the black lines in the ocean basins in the above map, are where you find hydrothermal vents, indicated by the red and yellow dots along the ridges. Hydrothermal vents produce high concentrations of iron. Here I'm showing you a section of dissolved iron concentration from a study that sampled downstream of a hydrothermal vent system in the South Pacific Ocean. The map on the top right shows the station map that corresponds with the section. The pink arrows show where the sampling transect passed over a mid-ocean ridge, and subsequent stations to the west sampled the elevated iron plume as water was swept away from the hydrothermal vent system to the west by prevailing currents. The region of the bloom we've been talking about is indicated by the pink box. And as you can see, this region is dissected, as I've already mentioned, by a mid-ocean ridge. In 2015, the year after we conducted our fieldwork in this region, 2 hydrothermal vent systems were discovered in the same area, and those are indicated by the pink dots. It's important to note that while there has been one previous instance of hydrothermal vents linked with a phytoplankton bloom, there is no previous work linking hydrothermal vents to a recurring bloom, like the one I'm describing here. So we're headed into some uncharted territory. Now we're back to the composite chlorophyll image of the bloom from November 2013 to February of 2014. I've shown you this about 100 times now, and I think it'll come up a few more times. So when we add the position of the hydrothermal vent systems discovered in the region in 2015, we can see that part of the bloom identified in the satellite chlorophyll image is positioned directly above the larger of the 2 vent systems. Overlaying the satellite-derived currents from December of 2013 shows that the bulk of the bloom and the section measured during our cruise falls downstream of the large vent system. When we look at the position of the hydrothermal vent systems relative to average net primary production from 1997 to 2019, we can see that the average position of the bloom matches suspiciously well with the position of the vents. So now we're going to talk briefly about hydrothermal vents, how they work, and what we think is happening before launching into another round of results. So hydrothermal vent is a fissure on the seafloor from which geothermally heated water discharges. When this water discharges, it is very hot and creates a buoyant, rapidly rising column of what looks like smoke. And this is because the water is full of particulates, like smoke. As this buoyant plume rises, the particulates in the plume settle out and ambient seawater mixes in, gradually diluting the hydrothermal vent effluent until it's no longer buoyant relative to the surrounding water, at which point it becomes a neutrally buoyant plume, which is then swept along by the prevailing currents at the depth where it reaches neutral buoyancy. As this neutrally buoyant plume is swept along by currents, it's gradually losing iron. I believe that some amount of the iron in the neutrally buoyant plume from the hydrothermal vents in our study region is mixed to the surface through the action of wind. I further believe that the shape of the bloom is determined by the shape of this neutrally buoyant plume and the elevated iron concentrations that it brings with it. To test this hypothesis, work was conducted by my collaborator, Sarah Sergi, using something called particle tracking. And this is where virtual particles are released into the surface current field, which we obtain from satellite estimates of surface current speed and direction. And these particles are tracked over a period of time, and their position is followed as they move away from their release point. Hundreds of these virtual particles were released at the surface above the 2 hydrothermal vent ridges near the bloom, and their progress was followed for 150 days. And this was done for each year from 1997 through 2019, using current data from November through April. So on the left, I'm again showing average net primary production from 1997 to 2019. On the right, the average results from the particle tracking analysis. As you can clearly see, when particles are released at the surface above the vents, the average shape of the bloom is recreated, further indicating the association of the bloom with the vents. The major conclusion from this work is that I have found a previously overlooked recurring bloom in the Antarctic circumpolar current, which I named the Australian Antarctic Ridge Bloom after the mid-ocean ridge with which it's associated. I further argue that this bloom is the result of a natural iron fertilization system that is fed by iron from hydrothermal vent emissions. So next, I'd like to talk about the implications of these findings. So now I'm showing you that same composite satellite chlorophyll image that I've shown you before, but this time the cruise track is also showing underway PCO2 data. The color bar for this is to the right of the image. As you can see, PCO2 within the bloom was drawn down to around 200 microatmospheres and lower, which tells us that this bloom has the potential to act as a carbon sink. So I calculated the CO2 flux within the bloom and got a mean flux of minus 32 millimoles per meter squared per day, and a negative flux is into the ocean, and a maximum flux of negative 131 millimoles per meter squared per day. By comparison, the CO2 flux in the Ross Sea in January, which I've mentioned previously is a highly productive region, is around minus 12 millimoles per meter squared per day. So we are seeing a CO2 flux in January of more than double what has been measured in the Ross Sea, a region that is considered to be a strong CO2 sink. Now, the region of this bloom is also consistent with areas of high krill populations identified by previous studies. So it's hardly surprising that humpback whales are showing an interest as well. Now, this figure shows the paths of whales tagged on the southeastern coast of Australia in October of 2008 and tracked through April of 2009. The color on the background of the figure shows the composite satellite chlorophyll image for the same time period in 2008 to 2009. Aside from a few individuals that took a little detour to New Zealand, here it's a lovely country, the whales travel pretty much straight south from Australia, hit this bloom, and start feeding. Finally, I want to summarize the main takeaways from everything that I've presented to you today. In chapter 1, I showed that net primary production has decreased with time in the Pacific sector of the Southern Ocean, along with decreases in sea surface temperature and open water days. I also showed that the Pacific sector shows a sub-regional response to the Southern annular mode. So we see substantial increases in sea surface temperature, open water days, and net primary production on the shelf, and significant decreases off the shelf during a positive phase of the SAM. In chapter 2, I identified a previously overlooked recurring phytoplankton bloom in the Pacific sector, which I named the Australian Antarctic Ridge Bloom. I also argued that this bloom is fed by iron from the hydrothermal vents underneath it, which is the first report of a recurring bloom linked to hydrothermal iron. Over the course of my dissertation research, I've been really struck by the importance of scale. The findings that I've just presented to you were possible because I looked at things on a finer scale. I'm not the first person to look at net primary production in the Southern Ocean or the response of sea ice in the Southern Ocean to the Southern annular mode. But for the most part, these studies have looked at the entire Southern Ocean. And when studies look at the entire Southern Ocean, they have to look at things with broad brushstrokes. However, when we look at smaller scales, we can find very important relationships in the details that aren't visible at the larger scale. And I'd like to thank you very much for your time and open the floor for any questions. I'm just going to pop that research takeaway slide back up. Thanks, Kevin.
All right, wonderful. So great job. And if people have questions, you can use the raise hand function and I'll call on you.
If you're shy, you can type it into the chat too.
That's right.
Can I just ask?
Sure, go ahead.
I put my hand up, but nobody, anyway. Hi, Casey, this is Dave. I don't know if you can see me.
Oh, hey, Dave.
Hi.
I can't see you. You're not popping up for some reason.
Great talk. So I have a bunch of questions, but I'll just ask one. So the Sam, I noticed that you went from what, 1990 to 2020 roughly?
Yeah,
The-
like 1992 to 2013.
Yeah, so and I don't remember when it exactly switched over, but it seemed like, even though there's a lot of variations, yearly variations, it seemed like we're now in mostly a positive SAM mode. Is that
Yes and no, because it's so variable, it still does go negative for sure.
right? But it seemed like it was more often than not relative to when you started. Well, the question is there any, have people looked at, gone back in time? Are there any paleo indicators that would indicate what SAM was in the past, and what might that inform us what might happen in the future?
You are very much heading into things that I am not sure of. Paleo stuff isn't really my thing. I imagine that people have looked at that. I mean, Antarctica is right there. People love to take ice cores there. So I'm assuming that they've looked at, but I don't know how you would get an indication in a paleo record of pressure systems.
Yeah, I don't know. I mean, they've looked at, El Nino, La Nina. They've looked at coral for oxygen. I mean, I don't know whether there is a tracer that could look at that, but I imagine there might be something.
That's a great question. I will probably do a little research and read up on that because now I'm curious.
And Kevin, if you don't mind, I want another really quick one. I noticed there was a paper by Clark in 2017 on iron isotopes coming from hydrothermal vents, and it's isotopically light, I believe. Have you looked at the isotopes to really pin down whether this you're suggesting that it is the iron from the hydrothermal vents is really what's fueling this?
Yeah. So we weren't able to look at any isotopes because you kind of have to want to measure them when you go there. It helps. So because we didn't know there were hydrothermal vents there, we didn't know to measure anything related to hydrothermal vents. So one of the big pushes that we're sort of making in the Auriga lab is to get a cruise to go back there to measure isotopic ratios. I think our focus is mostly on helium-3, less so on the isotopes, less so on the iron isotopes. My understanding, which is not very detailed about isotopes coming out of hydrothermal vents, is that it would be vent specific, that you wouldn't necessarily always see the same isotopic signature from a vent other than the helium-3 isotopic signature, which would be, I believe, light. Can't remember. It's different.
Yeah,
Certainly
they
different.
produce helium-3. You get, the helium-3 is a very good one. Anyway, it was a great talk. Thanks. I don't want to hog the questions.
I see Charles has a question. You got to unmute yourself.
So thanks for that very informative talk. We on Rose Lane are in applause of you. Okay. So my question to you, based upon what you discovered with this vent, would be what would you most be interested in pursuing in the future based upon what you've now discovered? What would be for you a next step?
Well, I think two of the big questions for me that came out of the work with looking at the hydrothermal vent as a potential cause of this bloom is my understanding of physical oceanography does not entirely account for how this iron, this hydrothermal vent plume gets to the surface so close to the vents. The work that's been done previously has looked at hydrothermal iron influencing a bloom pretty far away from a vent. And so I would be really interested in what happens between the vent and the surface that gets this. I have my ideas, but I would be interested to actually get there and measure that. And then I would also be interested in looking at the carbon export in a bloom like this. So how much of that carbon that's taken up by the phytoplankton actually makes it out of surface waters instead of being sort of recycled and eaten and all these other things that can happen.
Well, you touched on something that I did ponder as you were making your presentation, and that is there are these vents all over the world. So why in these two vents in particular, and there may be more, I guess that's the other thing that could surface, you could find more. but why does this seem to get to the surface and impact at a higher elevation in the column than one would, I guess, anticipate?
Yeah, there's a bunch of, I've got a list of hypotheses for why
Okay.
this makes it to the surface,
All
but
right.
it's also
All right.
a lot of the other vents throughout the rest of the world. I showed that big map and there's vents everywhere.
Right.
I think one of the really important distinctions is that these vents occur in a place that's iron limited. For the most part, hydrothermal vents aren't producing things that are needed by surface phytoplankton. So even if the influence of that vent makes it to the surface, you wouldn't necessarily see any
I
change
see.
in production
Right.
because
Okay.
it's not producing something that's needed.
Okay. Thank you, Casey. Well done.
Thanks for coming, Charles.
Any other questions from the general audience?
I addressed everything. That must be it.
Yeah. Seeing none, would you like to do your acknowledgement portion of your
Oh
presentation now?
God.